Good afternoon, and welcome to another Research in Action brought to you by the Division of Research at Florida Atlantic University. My name is Karen Scapinato. I work in the Division of Research at the FAU. It is my great pleasure to introduce today's speaker, Dr. Robert Kozol. Dr. Kozol is currently a postdoc fellow in the Department of Biological Sciences at Florida Atlantic University. If you have not heard what a postdoc is, a postdoctoral fellow is, that is a period of time after an individual receives his or her Ph.D. or doctoral degree, where the individual gets additional typical research experience before they move on to the next stage in their career. So Dr. Kozol is a postdoctoral fellow, and his expertise is in developmental biology, neuroscience, and genetics. He received his bachelor's degree from the University of Rhode Island and then moved on to the University of Miami, where he worked with Dr. Julia Dolmans. That's where he started looking into zebrafish mutant models of autism spectrum disorder and started working on, in general, on fish models, which are a relatively easy model compared to others to investigate diseases and how diseases occur so we can improve prevention and treatment of these. Dr. Kozol is currently in Dr. Eric Dubois' lab and has been there for about 2 years now, where he's creating novel cavefish brain atlases. And I'm going to have him tell you what that is all about. Welcome, Rob.
Thank you. All right, let me share my screen. So, thank you for that introduction. So today, yes, I have been trained and I'm very interested in how genes and differences in genes across different populations, whether that's a patient or whether that's wild populations of the same organism, how variation in those genetics shape the brain in both its anatomy and its function, and how that plays roles in changing behaviors. So today I'm going to really talk about these brain atlases that I produced here and the tools we've created to try to investigate how genetic variation leads to changes in brain anatomy, both in terms of evolution and disease. So one of the big burning questions in terms of developmental biology, in terms of neuroscience, is how do we get gross changes in brain anatomy, either in shape or in size? And so this is a good illustration of this question, which is if you look through primates, one of the primary characteristics of brain evolution is that the forebrain, this kind of colored, very yellow and orangish portion of the brain, is expanding until it's most of the brain when you get to humans. And so that takes a lot of changes genetically and developmentally, and we don't fully understand how that works. So part of our job as developmental neuroscientists and geneticists is to find model organisms in which we can ask the big questions, which are, what are the novel genetic types of functions that can cause something like forebrain expansion? And also, how could changes in genetics lead to things like degenerative diseases that change the anatomy of the brain, such as Alzheimer's or macular degeneration? And so we have many animal models to look at this. And these are our traditional 20th century animal models: the mouse, the fly, the zebrafish, also the worm. And this has been kind of our group of organisms that we've learned so much about genetics, developmental genetics, neuroscience. And as of recently, around 2012, we've started to introduce this Crispr Cast that you've probably heard of, that we can precisely mutate genes. And so if we know a gene is associated with something, we can cause it to be bad, let's say, and not do its function the way it's supposed to typically function in a normal organism. And then we can study the downstream effects. But that doesn't allow us to do gene discovery, which is to say, we need a way to look at two variable genetic organisms and ascribe new functions to genes that we otherwise don't know how they work or had been associated with different functions, but not the ones that we are interested in. And so we need something for gene discovery is what we're trying to say. And so that's why we use the blind Mexican cavefish here in the Du Bois lab and also in through our collaborative with Alex Keene and Joanna Kowalko, who just recently left the university to go pursue other labs. But the blind Mexican cavefish is a very intriguing genetic model because basically 28 to 30 cave populations sprouted from surface populations in the river system of this Mexican basin. And pretty much surface fish would be flushed down there, and over 100 to 200,000 years would turn into the pinkish fish you're seeing in the middle of the screen. And so it has a lot of defining characteristics of a cave organism. It's lost its eyes. It has no dark pigment anymore. Its metabolic functions have changed with a lack of resources. It no longer sleeps, is able to formidably deal with stress. So there's all of these interesting phenotypes. And although it looks like a different fish, both of these organisms still maintain biological fertilization. So we can cross the two organisms and get genetics from both populations to produce different phenotypes, anatomy, physiology, behavior, that we can then ascribe back to the genes that are found in the wild type cave and surface population. And just kind of talk about some of the big breakthroughs kind of in terms of disease and development in the last couple of years from cavefish. On the left, from Nicholas Rohner's lab, it's showing these graphs that basically just say cavefish are really bad at maintaining and dealing with high glucose levels. And they have all of the factors that on paper would say they're diabetic, but they're not sick. They're fine. They're living healthily for 40, 50 years in the wild. And so the question is, how do they maintain the signs of diabetes, but otherwise are not sick? So that's a big question that's opened up avenues for discovering breakthroughs in terms of diabetes. And then on the right, this is just showing you traces of fish swimming in a tank. And on the top, you see the surface fish who has just come into a new environment, is very stressed out and staying away from the top. But the cavefish on the bottom in red is swimming everywhere freely with no stress whatsoever. So they also have this reduced stress response and low cortisol levels. And so that's what the Du Bois lab mostly focuses on. And, you know, it's an interesting aspect that they're kind of superhuman in terms of their low stress or their ability to be diabetic but not sick. So this is why this is a very intriguing organism for studying biology in general, but also in terms of human health. And just to reiterate the amazing aspect of these fish to be a genetic map for finding novel genes and phenotypes, these are some surface to cavefish hybrid larvae. And as you can see, you get these myriads of phenotypes. So some will have bigger eyes or smaller eyes. And that's going to be due to the genetics coming from the parents that are either surface or cave. There's pigment cells. So in the top two, you have pigment that's being generated. In the bottom, you don't. And even in the eye, you can see is not getting those retinal pigment cell layers that you need for vision. So this illustrates the idea that we can have a mixture of the genes, and then later on, we can try to find the genetic elements that are responsible for the differences in anatomy, physiology, behavior. So really today's talk is going to be how do genetics and development impact brain-wide anatomy? And again, this is a very important topic, and because we're using a fish that is small and see-through at a larval period, we can actually look across the entire brain, ask how individual brain regions change between the two populations, and also how all of those brain regions interact together developmentally to affect the size and position. And so the only way to do this is to have an atlas. So as I alluded to earlier, basically this is similar to any kind of atlas you'd have geographically. There's different positions in the brain and they can be segmented into little functional blobs. So you're looking at this zebrafish atlas with all these colorful little positions on the brain. And each one of those is a neurodevelopmental region that is either molecularly or functionally characterized. And we can say that region is X, it's involved in this process. And so once you have a brain atlas and you're able to sample a lot of fish, you can ask what's changing anatomically if you know there's difference in genetics, or perhaps what's changing physiologically if you bring in some other analytic tools to look at activity in the brain. So today we're going to focus on anatomy. And so most common animal models have brain atlases and these brain-wide tools. And so when I came in, I helped to develop some novel atlases so that we could look across all groups, the cave, the surface, and the hybrids, and some anatomical tools to compare both the shape and the size of all brain regions across these three groups. And so this is just showing you the process of how we did this. So this is kind of the methods, which is we stained these fish for a neuroanatomical marker, the one that's in black that you're seeing. And so that's giving you an outline of kind of the architectural highway of the brain, the neuropil regions. And so these are the regions that are the axons and the dendrites that are sending the signals from neuron to neuron. And so this gives us a nice outline or a scaffold of the brain. And so what we do is we use a registration program that is able to overlap our brains onto the zebrafish brain. And so this would be the same concept if you went and got an MRI of your head and you wanted a doctor to analyze that. They would have to overlay your brain scan onto a template where they know all the brain regions and they can then analyze them for size or if it was activity, et cetera. So the good thing about this is we can use the instructions of registering our brains to the zebrafish to reverse the maps in zebrafish onto our cavefish. And then we can validate that in different ways to say whether or not it's accurate. So we went through that process to create this, to utilize kind of 20 years of zebrafish neuroscience research to then borrow that and bring that into our model. And so we iteratively did this process for tons of surface and cave individuals. And then this is showing you a bunch of F2s. You just go through, you register the fish's brain, you bring the map back. And then what we wanted to do was be able to look to scale through the brain. Look at the whole brain, then the four major regions. So this includes forebrain, midbrain, hindbrain, and then 26 more functionally defined units, and then the 180 from the zebrafish, which provides kind of a fine scale street view for your ability to look. And so the first thing we wanted to do was just look at how surface and cave size of brain regions compare. So this is without the hybrids. And so we'll start just by looking at one of these regions and scaling through and seeing the kind of finite detail we can get in terms of anatomical differences. So if we're looking at this portion of the forebrain called the diencephalon, you see it in purple. And here, if you look at the graph, really what you're looking for is whether these blobs are larger or smaller than each other. Are they higher or lower? So here you see the cavefish group is higher, so it has a larger percent of total brain volume for this region. So it looks like this portion of the forebrain is expanding in cavefish compared to the surface. But that's something we've kind of already known. So we'll go lower and we'll say, let's split it into two. One side's the hypothalamus, one's this dorsal portion. Here, we find something we've also seen in the literature, which is cavefish hypothalamus is larger, but we have a novel finding, which is this dorsal portion is actually larger in surface fish. So that's kind of the excitement of this brain-wide process, is we get to reaffirm and test results that have come and have been generated in the field of cavefish. And then we also get to see these novel things in areas that nobody's looked at. And then finally, if we try to go for that street view, we get more novel findings, as in the back portion of the hypothalamus with the intermediate and the caudal. They account for the hypothalamus expansion, not the preoptic and rostral. And then if you look on the left side, we now know this little functional unit called the dorsal thalamus is bigger in surface fish. So the cave have reduced the size of that thalamus. And that's important, especially if you want to study motor and sensory relay between the cortex and other regions. And so the thalamus is important for understanding things like Parkinson's and ataxia and visual disorders. So now our question would be to build hypotheses for what those changes in shape, how they might be affecting behavior. So how do genes relate to all this, right? We're just looking at wild type populations. It doesn't really tell us anything. It just says they have differences that could be interesting. So one way to look at it is to look at the hybrid population in comparison to your wild type. And that is, if we see hybrid characteristics that look like either surface or cave, then we can say there's a high likelihood that genetics from those populations play a role in the development of this region. So here we're looking at the midbrain, so the mesencephalon. So that's that green splotch on the surface in the cave. And this doesn't look very interesting. The hybrids just seem to have a smaller midbrain. They seem very much like the cave. So then if we break this up again, and we start scaling, we see that that's all due to this large visual center called the optic tectum, that kind of ball that's on top. And so if you see the red arrow, it's showing that the hybrids look more like cavefish just for this optic region, and otherwise the tegmentum is relatively unchanged across all populations. Now, the part where the hybrids become very instructive on how genes from the wild type surface or cave might influence behavior or influence anatomy come into play here when we get to an area of high variability. So although the white matter, again, this is kind of the highways, the areas that are sending signals from neuron to neuron, look in terms of the hybrids like the caves. On the right side, we can see that we have this long range where you have this top portion where a bunch of organisms have a very surface-like gray matter or cell layer region within the optic tectum. And then the bottom portion looks more like cave. And so this is where we're going to be able to untangle genetic links to how these changes in anatomy through development have occurred. And so again, if we break this down, we can get a much better street level view of what's going on. And again, if you look to the right to that green section, this ventral portion of the tectum, we see the top portion of animals with a higher number look like surface and the smaller number like cave. And so what we do now is each fish was individually imaged and went through the experiment, and we've collected their DNA. So now their genomes will be sequenced, and we will link and associate variations across the genome with these types of features as being either surface or cave. And so this is interesting in terms of individual regions that we might be interested in behavior. But one of the big developmental questions is, how do these changes in brain segments and size relate to each other? And so my mentor, Eric Dubois, one of the things that he was able to do is do this correlation experiment. So really we wanted to find associations in volume between brain regions across the entire brain. And so don't pay attention to the numbers so much in this, but really look at those colors. Because what this is saying is, if you compare the size of the tectum, that kind of ball that we were looking at, that visual sensor, with the hypothalamus, that lower brain region we looked at previously, is that when one gets bigger, the other one gets smaller. So if you see something that's very blue in the block, they're negatively associated with each other. So this is comparing all of the hybrid individuals up to this point. And it says that, yes, the hypothalamus gets larger when the tectum gets smaller. And basically, again, this gives us an interesting interaction between brain regions, but it doesn't tell us what's really going on developmentally. And so to look at that, we need to do what is a cluster analysis of this data. And so by doing a cluster analysis, we start to associate groups that kind of move together developmentally. And what we find is we get these two regions that are kind of positively associated with each other. So everybody in that blue circle, as one gets bigger, the other gets bigger, they're moving in concert. And everything in the red group, again, is moving together positively. But they also are inversely correlated. So as one gets bigger, as these large S groups, the other one gets smaller. And so this is kind of hard to look at a graph full of colors to understand, but I think it makes more sense when we look at it developmentally. So if you look at cluster one, they're all in this ventral portion, really down low in the brain. And if you look at cluster two, it's all in this dorsal portion. And really developmentally, we know these regions are linked together early in development. They have little organizers that have gene regulatory networks that are saying who's going to become who and how to grow and how large to grow. And if you start to change the genes in these regions, they're all going to change in concert. And even before that, you also have time periods when both regions are going to be impacted by large-scale genetic changes. So we know if you start to impact the genes that are coming from an organizer center or previously, you'll start impacting the anatomy and the development of the brain. And so now this provides a nice overarching model for what might be going on to change the size of these brain regions in the cave fish. And luckily, a lot of these hypotheses have already been around, and we are either lending support for this kind of early gastrulation time period that could be really changing the genetic developmental landscape, or a little bit later on when you have these local centers that are telling these regions how to become and how large to become. And so again, now we can go back and say, what time periods do we know what genes are working together, repressing and activating each other to cause larger anatomical changes? And so this is just showing you as you go through in time, genes become, more genes come into play, they become more restricted, you get these specification of areas. And so now we can pick a few developmental time points and kind of look at some of the genes we're interested, and also, once we get the genetic information, ask what's going on with these genes in terms of associating with a cave phenotype of a size of a brain or a surface phenotype size of the brain. So now we've got this to go back to kind of the thing we talked about in the beginning. So how do these innovations for brain expansion, midbrain contraction, how does this all contribute to behavior? And so this is kind of the next part, some of what I've done already and what we're going forward with. And finally, what are the genes that are influencing this anatomy? So one of these simple behaviors that I've been interested in, because a broad question in my research interest is just how do fundamental behaviors exist? So how does a behavior change, or how does a novel behavior evolve? And so you need genetic changes that lead to developmental changes that change anatomy or physiology, and that the output then is a difference than what you saw before. And so this is a good example of it. This is a phototaxis behavior. So if you look at the diagram on the left, there's a little light box and a camera above it, and the light box just goes on and off every 5 minutes. So if you look at the graph, the white bars below on the x-axis as the light is on, the dark as the light is off. That repeats on cycles for 40 minutes. And pretty much we just record how much movement the fish starts doing. And if you look at the black line, the surface fish, they're very active when the light is off because they're positively drawn to light. So they are searching for the light. And when the pichon has the light on, it's positively searching for the dark. So it's scototactic. It is looking for dark. While the surface is phototactic, it's looking for light. So this gives us a nice binary system to where we can say, okay, if we transform this data, if you're a surface fish, you're in the positive index here on the y-axis. And if you're a cavefish, you're in the negative axis. And what we see with the F2s is you get a mixture of both. So again, we have this behavior that's quite variable and binary. And when you look at it in hybrids, you're either surface-like, pichon-like, or neither. And so this is one behavior where we can use this anatomical data that we're collecting and also some physiological data to ask what brain regions are responsible for this change, and also what are the genes that impact this using the F2s that we collect. And so on top is just showing you one way you can do this physiologically. These are brain maps of activity. The green portions are showing you what brain regions are active when the light is on, and the magenta regions are just showing you what brain regions are active when the light is off. So we're able to use that, just like the anatomical data, to find the little brain segments that are active and the neuronal clusters that we're interested in studying. And so that's currently what we're finishing up with the physiology aspect. And then really quickly, I just want to talk about, the big thing is when we do the genomic work to find the genetic variants that underlie these anatomical differences. But in terms of behavior going forward, we're not just going to be looking at sensory motor behavior. We're very interested in the forebrain expansion data that we got from the anatomy. And we really want to use this to look, we're already looking at it for stress, which is, again, a major portion of the lab. But I'm also interested in variations in things of memory and learning, because the regions that have really gotten much larger in the kind of dorsal forebrain are important for things like memory and learning, and also emotion. So we're starting to develop behavioral paradigms so we can study that in relation to either disease or evolution. And so with that, I'd just like to acknowledge my mentor, and also Harry Burgess at the NIH, who helped us create these atlases and these tools. And then everybody in the Du Bois lab, the techs and the graduate students that have helped out with this project and that I helped mentor. With that, I will give it back to you.
Great. Thank you very much, Rob. Just as a reminder, if you have any questions for today's speaker, please use the Q&A button in the bottom of your screen and type your questions there. We will go through them as they come in. So Rob, fish is a relatively easy model to do some work on, as and you showed some of the things you can do with the fish. Can you talk a little bit about the limitations, especially when you're studying human disease, on the limitations of a fish model?
Yeah, I mean, so there's a few kind of specific to fish limitations. One that's very glaring is there was a duplication event in the genome that happened after fish radiated from the other vertebrates. And so there's a lot of times, especially if you're studying neuropsychiatric disorders, the genes will have two versions, which we don't in humans, we just have one. And so then you have a whole complex array of how those genes now function. Did they break the functioning up, one for one gene, one for the other? And so that's just one. And then otherwise, there are obviously things that you just can't study. Vocal communication in zebrafish, so it's limiting for certain things. When I was studying autism, so you might focus on more social motor. But certainly, as much as that's limiting, the access to the nervous system in freely moving organisms is definitely the most powerful aspect of using any kind of fish model, in my opinion. So.
And you mentioned, or we mentioned earlier, that zebrafish is what you started with, from what I understand, doing your graduate work, and now you moved on to cavefish. What's the difference between those two models, and why one versus the other?
Well, really, and that's why I tried to kind of expand on 2 or 3 slides about the hybrids in the beginning. We don't have many hybrid models that we have a sequenced genome for, that we can be able to take the hybrid, find an interesting aspect of behavior or anatomy or physiology, and then go back and say, can I find a gene that's associated with this type of feature? You can't do that in zebrafish because they don't have enough genetic variation that you would have 2 populations that are so different in terms of genes that you could go back and look through the genome and statistically find something that's going to be correlated with those features. So really it's the hybrid aspect, but then also the cavefish, like I said before, because they've had to develop in such a unique environment, they have diabetes without being sick, like very specific or something that my boss studied in when he was doing his PhD, and what Alex Keene took over from NYU from there, which is sleep in cavefish. Like cavefish don't sleep. And we are pretty sure sleep is fundamental biologically for pretty much the majority of organisms. So kind of taking sleep out of your repertoire is very confusing. And that's helped us understand aspects of sleep and sleep rebound. So
I would like that if I could give up sleep. I had a lot more hours in the day.
Yeah.
So you mentioned identifying genes that are associated with certain diseases. How likely is it to find one particular gene that, or even a mutation in a gene that is actually associated. And you mentioned you're working with autism. So is there one autism gene that may be mutated in some humans and not others, or is it a combination of things?
Okay, so I guess is this two questions? One would be the first is just like how do you find the genetic variants in the cavefish population? And the next one you're asking kind of in terms of autism, how does the genetic landscape work clinically? Like
Sure.
what are the okay? So the first one, pretty much it's the majority of differences genetically, at least that we think in cavefish, is actually changes in gene expression. So it's not that you'll find that mutation that changes, that makes the gene different in terms of the protein, but that actually where the gene's being expressed is changing. And so you find differences in mostly non-coding regions. And when you do these kind of QTL mapping of trying to figure out what genes are associated with what type of feature, a lot of the times you're going to get a bunch of genes and you're going to say, okay, now we have a list of X amount of genes. What do we know about them? Where are they expressed? And then you can generate your hypotheses and then functionally test them. Sometimes you get lucky. Obviously, in cavefish, there's been a couple studies where doing that method, they found a single gene, and that single gene causes loss of pigment or degeneration of the eye. And so there are those cases, but more likely you will get a list, and then it will be your job to do the testing to find out whether or not those are responsible for it. In terms of autism, autism genetics has been kind of a difficult road for the genetics community over the last couple of decades because early on the assumption was that we'd find a couple of genes and they would explain the majority of cases. And really it was the opposite, which is you found a few genes that made up 0.5% to 1% of the population for just, maybe a 10 to 12%. And then there's anywhere from maybe 30 genes that we know likely are causing autism. And then you start to get into 100 genes, 400 genes that have these really rare cases, and we're pretty sure they have to do with autism. And so the genetic landscape, we know genetics plays about 70% of a role in causing autism. And then there's this 30% that we think is environmental in other cases, but it's been a harder, I think, than the community thought a few decades ago to genetically pin down autism. So
Is that because it's a combination of variations in genes, or is it a number of different diseases like cancer, for example? It's really a number of different diseases. It's not just one disease because you can have variations in a variety of different genes. So is autism because it's a combination of changes in different genes that come together, or is it a variety of different diseases really that affect different genes?
So certainly in terms of the way genes interact and the brain specifically, you have all of these gene and protein networks that, a lot are synaptic or some are within the cell body. And so we've done a lot of molecular genetics in the autism world to show that you're right. There's dozens and dozens of genes, and they could be misexpressed or they could have a coding mutation. And then in terms of how severe those changes are at the developmental and then later functional level, this is why, autism is a hard disorder to pin down clinically, because there's such a spectrum, which is you could have low functioning where you have difficulties cognitively, so you have problems with learning and just being able to speak, and then you could be on the high end and just have social deficits or communication deficits, but being of average or above average intelligence. And so, each one of these is a manifestation of these different genes being expressed in different ways at different time periods. Did you get affected very early on? And again, this kind of plays back into my talk today, which is when is the gene changing expression how early and global is that going to affect things? How late and compact is that going to affect things? Is it just functional once your brain develops? So that's kind of the difficulty. And certainly breaking a disorder up into multiple layers, one with communication, as in verbal, nonverbal, one with social interaction, one with sensory motor deficits, and then checking off boxes of each one of those and then telling somebody they're autistic. I mean, that's a wide range of things that can put you into a category. So yeah, I think it's just a quite a complex disorder on the genetic, developmental, and then behavioral, scale.
Sounds very, very complicated to really tease out. So it sounds like the fish model will really help there because it's also a relatively quick model, right? Can you talk a little bit about the CRISPR Cas methodology in case our audience doesn't really know and is not aware of what that actually does and how it functions?
Yeah, so actually, and there's a lot of CRISPR work happening right now. Like I was telling you before, there's different ways we get these gene lists. Like as I was discussing, when you get back to the genetic level and you go, okay, I've got these 20 genes I think are interesting for this difference between the cave and the surface. And so now you've got to do 20 functional experiments. And so now the beauty of it is we can finally say, okay, I want to stop this gene from being expressed. So I am going to take this little basically complement to your DNA. It's going to have the exact same letters to it. And then I'm going to attach this protein onto it, and it's just going to break that strand. And so you've got your gene, put your little thing on it, it breaks it. Now it's broken, and it's not that it caused a mutation, it just broke the strand. Then your cell itself goes to put it back together, and it makes errors, and it accidentally puts in letters, and it takes letters out. And that's how we disrupt the function. It's really hijacking your cell to make proofreading errors. And then once we do that, if it's injected into, a fertilized embryo, hopefully we'll get these mutations across all the cells. And then we can say, what happens when you grow up without this gene function, when you've disrupted this protein? And so we're actually doing that. Last night, probably 4 people were in here injecting embryos because the cavefish spawn at 2, 3, 4 o'clock in the morning. So they have to inject at that point. So there's a bunch of people that aren't here right now. They're probably sleeping. But they were doing that, injecting dozens of guides and some constructs for me as well. And so, yeah, that allows us to disrupt gene function and then go back and say, hey, are they sleeping well? Or are they, can they sense the light? And that's how we do that work.
The life of a graduate student, right? To work any hours of the day, which gets me to our next question here. How did you become interested in this work, in particular working with fish and fish models, and then also working on autism spectrum disorder?
Yeah, well, the fish came first because I went to school to be a marine biologist and a deep sea ichthyologist. So I wanted to study cool things with fish in the Marianas Trench. And then when I needed a job after the Great Recession and was able to get one down in Miami, I was fortunate because it was for zebrafish with Julia Dahlman's lab. And it was just, hey, the med campus has a grant. They want to test a proof of concept that you can make a autism gene model on zebrafish. And then I started learning development in neuroscience and became a neuroscientist that just happens to work with fish models. So.
Great. Now you're still in your education, so to speak, right? Yeah. You're out of your Ph.D., but you're still having to look for your next position and gaining experience right now where you are and the position you're in. What is your next step, Rob? Where do you see yourself going?
Oh, well, yeah. So next year I'll be on my first job market. Sometimes it can take a year or two. So next year I'll just apply for any open professor positions that are looking to have an assistant research professor, and really only limited to, well, I'll be looking at broad where they go, we just want a biologist, all the way down to, we want a evolutionary biologist or a sensorimotor biologist. And so probably applying at a bunch of schools next year and hopefully starting my own lab. And right now it's really just continue writing grants, and this atlas tools and morphometrics and stuff that will be published soon. And we're going to be doing a similar thing, like I said, with physiology. So I'm really going to be bringing the high-tech world of zebrafish into kind of the zebrafish world, which is just it's a later model, so it's behind. And so hopefully that will be kind of my in, which is really developing this whole brain analysis field in cavefish. So
very, very interesting. So you're looking for an academic career now, and you mentioned publications. So, as a researcher, you have to publish, but you also have to find funding, right? Which is not always easy, especially not for research that is not easily understandable to the public. Can you talk a little bit about how research like the one that you're doing is supported?
Yeah, I mean, well, it's the traditional route through NIH and NSF. So with the NIH, I mean, the main thing is if you have some tie into understanding mechanisms of biology that underlies disease at any level, you just have to make sure you are making that argument in your funding opportunity. So an example is recently we did an F32 to get another year of funding for my postdoc. And really the way I was writing that is we know based on some mutants that the eye plays some role in this phototaxis change. And we know that that incurs degeneration of the eye. So we're now saying, we have a behavior and we have an eye degeneration model, and retinal degeneration is, a pretty big problem. And we don't fully understand what happens during retinal degeneration across the brain. So now we're able to take these different fish, either hybrids or mutants, that might have smaller eyes or no eyes or big eyes, and ask what's going on outside of the visual centers. And so with that grant we were writing, there's different ways we can now look at what happens when retinal degeneration happens anatomically and physiologically across the brain. So there's different ways like that where we really want to know the basic biology, like how does a brain anatomy, how does it change? How do you get forebrain expansion, all this stuff. But those all are very important for, developmental and degenerative disorders. So we're talking forebrain expansion. Part of the hypothesis out there is as we've also expanded the brain, it's caused problems when we get into old age, like Alzheimer's and things like that. So that's also a question of once you get to this higher level, like how does the genetics and development create neuropsychiatric disorders, et cetera. So again, you need to make sure you're posing your question correctly and writing your argument cogently.
Just as a reminder to our audience, if you have a question, please use the Q&A button at the bottom of your screen and type your question. Audio is not turned on because of the number of attendees we always have. Coming back to funding now, trying to get funding through NIH or NSF is not easy, right? The success rate is somewhere in the 10 to maybe 20% range, if it is. Yeah. Yes, it depends on which mechanism. And it also takes a while, right? I mean, if you're submitting your application today, you may know if you make it through the first round, you may know maybe in a year or so you have the money. So it's never easy. And maybe an appeal to our audience here, if you have an interest in supporting this type of research, please let us know. We'd be happy to get you in touch. Okay, I think we're out of questions here. So if anybody retrospectively has any questions for our speaker today, please feel free to either contact him yourself or contact me and I'll get you in touch. Again, a recording of this presentation will be available on our website, FAU Research in Action. And we will be back next week at 1 p.m. again, Eastern Time, to talk to you about our next topic. Thank you very much, Rob. It was a pleasure listening to your research. Good luck with your career and your career path. Everyone, have a great rest of your day. Bye.
Bye-bye.